Hey guys, welcome back to Bioinformatics, Computational Biology. In this lecture, we're going to deal with unannotated transcribed regions. So what does that mean? So in some cases, you will have transcripts that map to part of the genome that is not annotated. So we don't know if it's a gene, what the gene does, what the name of the gene is, anything like that. And so this happens a lot of times if you have poorly annotated genomes, so maybe you're not doing a model organism like Arabidopsis or mouse or C. elegans or something like that, and instead you're using a more novel genome, either a genome you constructed or a genome that just doesn't have a lot of annotation to it. So, or you could be just looking for genes that are not annotated, such as in Arabidopsis. Maybe there are genes that have been missed and you've had a novel experimental condition that caused this gene to be expressed that has previously not been annotated. There's a lot of hypothetical proteins and things like that in the Arabidopsis genome that have not, that these gene prediction models have suggested that we don't know much about, and so there's a possibility that you find another gene that hasn't previously been discovered yet. So we're going to look at how to use R to map transcripts to unannotated regions. So let's get into that. Okay, so first things first, we are going to load some libraries here. So the first one is locfit. And the second one is our SAM tools. So let's run those, get those loaded. Perfect. So let's create a function that will load a gene region, or load, I should say, the gene region information in a GFF. So a GFF is a gene features file, and that has all the start and stop locations and exons and introns and everything if you're doing a eukaryote for this genome. So a GFF file, and convert it to a bioconductor GRanges object. So this is kind of like a nested or a matrix file. So our function is going to be get annotated regions from GFF, and our function will be called, or function will ask for the file name. Then we will make an object called GFF with the rtrack layer function, and from that package we'll take the import.gff function for whatever, and run it on whatever our file name is, and then make a GRanges object. Okay. So also, this using GFF files, we're going to use that, but you can also use bed files. If you look into the this rtrack, it should be layer, not later, rtrack layer package, you can see what type of files you can input. So we're going to use GFF, but you can also use bed, and I think there might be another one or two. Okay, so we're going to get counts in Windows across the genome in 500 base pair segments. So actually I'm going to run this function to load it up there. Okay, so now what we're going to do is we're going to map the counts, so the actual mapped RNA transcripts, and we're going to look at 500 base pair windows across the entire genome and see where those counts fall into those bins, right? So we'll call this whole genome, and we're, whoops, it's not the right arrow. And we're going to use the seesaw window counts. So as you can see here is count, convenient function to compute filter statistics for windows based on proportions or using enrichment or background. So this is going to kind of do our statistics for our windows. And so we're going to use file path. So for you, you're going to use file path, and then it's going to be desktop, classroom, and then my files. If you're using Praxis. For me, since I'm on my computer, or my desktop, file path, get working directory, for the data sets folder, the chapter 1 folder, the Windows and then the Windows.bam file, which has our counts in it. So we're going to bin them up. Filter equals 0. Width of our window, you can custom set it. We're going to set it to 500 windows, 500 base pair windows across the genome. So parameter, we're going to say seesaw, read param. Okay, so now we're going to set the parameters for what we consider to read to be mapped. So min-q, this is the quality of the read. So when we do the actual mapping of reads to the genome that you would do on a high-performance computer, there are Fred scores, they're called, which is a way to say how certain that is mapped to the correct location. So I'm going to annotate this. Determines what is a passing read. So what we're saying is that we're cutting off transcripts that mapped with poor scores. So if it's not, it tries to map it, it's like, I'm not 100% positive that this goes in this location. If it's under our certain threshold, which is a Fred score of 20, then we're going to throw them out. Dead up equals true, and this removes PCR duplicates that can arise as part of the mapping or the mapping process. And PE equals both. So this is this requires that both pairs of reads are aligned. So during the process of mapping, you can either use single end reads, so you read in a direction one way in the gene, or you can do paired end reads. And so it's just two different types of sequencing techniques. Paired end reads are better because it's just more thorough data and it requires these matched up. And so we're going to use, this data has paired end reads, and so we're going to use the paired end reads equals both function. All right, since this is a single column of data that's generated, let's rename it. So actually I should probably run, I ran that function, so let's run this here real quick. Error. Where did I put something that didn't need to be there? So that's chapter 1. Right here. Got a comma. Okay. Oh. Session. So I'm not in my right working directory. So let me quick choose my working directory. And informatics, VDB scripts, Annotated Regions. Okay. So let's run this again. There we go. Perfect. Okay. So now you, let's rename our column names for whole genome. And we're just going to call this small data, because we're using a small genome. Okay, so if we look at whole genome, you can see there's all sorts of row ranges. It's one of these nested matrix type. It's a GRanges file. So let's do annotated regions, and we're going to get annotated regions from GFF. So this was our function that we made up at the top there. And we're going to do file. You're going to use your file path, the same one up here, because it's all going to be all your data is going to be in the same file. But I'm going to use this function to read my files in. That's called genes.gff. So remember, you're going to use this file path right up here, if you're on Praxis. Okay, so let's run this real quick. How about I not import? Right. Something up here. Get annotated regions from GFF. Okay, that's right. File path. Get working directory, comma, data sets. Chapter 1. Genes.gff. Oh, it's because up here I say import instead of import. This is a lot of what you do in our, oh, we gotta run it now, is debug. So, get used to it. Okay. Alright, so annotated regions we got now. So it's a, now this is a GRanges. The previous one was a ranged summary, summarized experiment. So, and they're quite similar. They just have their data slightly arranged differently. All right, so now that we have the windows of high expression, we want to see if any of them overlap with annotated regions. So right, so here we did counts over in those 500 base pair windows over the entire genome, right? So you take the entire genome, break it up into 500 base pair chunks, and then determine how much expression there is at those chunks. Now what we're going to do is we're going to take the entire genome, and we have these gene GFF files, or gene GFF annotations. So it's taking the entire genome. It says this region's a gene, this region's a gene, this region's a gene, this is not a gene, etc. And now we're going to see the overlap, right? Where our 500 base pair chunks overlap with annotated genes. So we're going to load another library called GRanges. And then we want to load another library called Summarized Experiment. And we are going to find the overlaps between the windows we made. So let's do windows in genes, and we're going to say GRanges and use the overlaps any function from the iRanges package. And we're going to say summarized experiment row ranges of whole genome. And this part here creates a GRanges object. Annotated regions, and then let's close it off. Okay, so let's. I have too many parentheses here. I may. Okay, let's run this. Okay, so window in genes. So what this does is it goes over the genes, and using that GRanges object, let's print it out and see what we get here. Maybe I need an N there. It goes over the entire thing, these windows, and says whether it's true or false that it overlaps. So now what we can do, if there's an overlap, I should say, between our window and that annotated region. So now here we subset the whole genome object into annotated and non-annotated regions. Okay, so let's split off now the ones that overlapped with previously known annotated genes or regions and the ones that didn't. So let's call this one annotated window counts, and we'll do the whole genome for windows in genes. Okay, so annotated window counts. Okay, so this is where we had the overlap, and now the non-annotated window and counts is equal to whole genome. And remember from our previous kind of basic intro to R, if we put the exclamation point, it means do the opposite of. So windows in genes. So this is going to create everything that's not annotated, right? The opposite. Okay, so we have both those now. So now we're going to use assay to extract the actual counts from the GRanges object. So, right, so we have these GRanges objects, and everything's like kind of buried in here, right? And these are always kind of hard to understand what's going on in here because they're like nested matrices, right? So we can use assay though to kind of pull something out. So we're going to say assay for the non-annotated window counts. And you can see that we have these two windows here that have no annotation and counts mapped to them. Okay, so in this step, we use our SAM tools, pile up function to get a per base coverage data frame. Each row represents a single nucleotide the reference count in the count column gives the depth of coverage at that point. Okay, so what we're going to do here is we're going to load another library called Bump Hunter, and then create a data frame called pileup data frame, because we're using the pileup function, just easy to remember. And use our SAM tools pileup. And then for you, you're going to use your current directory, right? So you use that desktop classroom my files. And then for me, I'm going to just copy and paste this right here. I don't want, let's see, but we actually want to use the BAM file, which has the counts instead of the gene annotation file. Okay, so let's run this. Pileup PDF, perfect. So let's look at this. Okay, so we have, we're just doing the single chromosome, I believe. Let me double check. We're just doing chromosome 1, yeah, it's just to make it less data. And so this has the position, the nucleotide, and the counts for that nucleotide. Okay, so this step groups the reads with a certain distance of each other to a cluster. We give the sequence names, position, and distance. Okay, so when you have a transcript read, the reads aren't necessarily, you have a gene that's really big, and reads are only 125 base pairs generally. We'll assume that's the type of sequencing they did. So you have reads that can overlap, but they don't 100% overlap, right? So only like, you know, the first 25 from this read and the first 25, or in the last 25 from this read don't overlap, okay? So we want to group them with the ones that have overlap together. So let's do clusters from Bump Hunter. Oops, I didn't run Bump Hunter, did I? What? No library called Bump Hunter. Alright, let me quick install it. Thought I got all these installed. Okay, let's run this. This again. Okay. There we go. Bump Hunter. Cluster. Oops. So genomic locations are grouped into clusters based on distance. Locations that are close to each other are assigned to the same cluster. This is performed for each cluster independently. So, cluster maker from our pileup data frame. Seq names. Pileup data frame position, and our max gap. So genomic locations within the max gap are from each other are placed in the same cluster. So if we look at that file that had all the base pairs, it's going to group things close together in 100 base pair segments into one cluster. it's just piled. It's called a pile DF. Probably should have called it pile up. Okay. Clusters. Okay. So now what we can do is we have our clusters object, and we can just run table of clusters and see what we get. Okay, so we have these 3 clusters here that we've identified. Now let's run bump hunter region finder for pile up, or no, I just called it pile, right? Pile data frame. Counts. Count. Just count. Pile data frame. Seek names. Pile data frame. Position. Clusters. And our cutoff. Is going to equal 1. Okay, so actually I should annotate this, right? So scroll down here. So in this step, we will map the reads to the regions we found for the genome. There's no R or A in region. Okay, so this is what we end up with. So you see we identified 3 regions when we ran our table clusters here. We identified 3 regions that were not 3 areas where transcription mapped to that were not annotated regions, right? So these could be genes that we didn't know were genes before, okay? And so then what this bump hunter region finder does is it takes those and it takes our information from our pile DF, and then it says, okay, region 1 is on chromosome 1, and it starts at base pair 500 and it goes to 1500. And here's kind of our statistics for how they mapped it or whatever. Region 2 is from base pair 2501 to 3500. And then region 3, that mapped reads, that wasn't a gene, is from 4500 to 5500. So from this, if you, like I said, if you have, if you're curious about a genome that you think transcripts are arising that don't map to something, this is a way to map those RNA-seq transcripts to unannotated regions. So like I said, this has some use. I've actually never, I use Arabidopsis a lot, and Arabidopsis is I should say definitely, the most annotated genome for plants. If I were using, I might run this on some of my data just to see what happens after I, taught you guys how to do it. There's not enough time in the day, but it could be useful if you are using, doing a different model organism, or maybe you get lucky and you find something that wasn't annotated before, that is novel, important, exciting. But it's a good tool to have. We did this, 30 minutes. Wow, this is a lot longer than I thought. So that being said, I'm going to cut it here. I could talk forever. I drone on and on. But I hope you enjoyed the video. I'll catch you in the next one.